Hello, welcome everyone, and thank you for joining us for our first webinar in 2020, Epigenetics, How Your Genes Express Themselves, presented by Dr. Robin Murphy, naturopathic doctor. My name is Dr. Paul Herkel. I will be your host for today's presentation. Before we get started, I would like to go over a few housekeeping items. There will be a Q&A session at the end of the presentation, time permitting. If you would like to ask a question, you can do so throughout the presentation by typing it into the question box in the control panel at the right side of your screen. Please note this presentation is being recorded for future distribution. We are now ready to begin, so without further ado, I'll turn over to Dr. Murphy. Welcome, Dr. Robin. Thank you so much for joining us today.
We're hearing a lot about this, mainly because direct-to-consumer testing, genetic testing is available. This is in part because of the decrease in the cost down quite substantially. And with this, we're also seeing a rapid increase in the amount of research in understanding exactly what these genes do, how it affects our health, and importantly, what we can do about that. So I'm excited to share with you guys today really some nuances about not only how our genes get turned on and off, some of the differences between epigenetics and some of these terms that people are using on a regular basis, and then genetics and how those differ. And then more importantly, how we modify our genes. So it's not just about how these genes get turned on and off, it's where do we focus our interventions? So we'll talk a little bit about how this applies to detoxification pathways, and then looking at how to direct and modify gene expression using particular substances. And I think this is really exciting because we can get into the kind of the world of anti-aging, where we're actually modifying how the genes are getting expressed to affect the aging process and the cells and how they function. And then finally, we'll get into lifestyle and dietary factors and some of the nutraceuticals that there can be a risk for deficiency, as well as how to compensate for that. And then how do we use this information? So who benefits? How do we apply this into clinic? And where do we go from there? So nurture your environment, how you grow up, your environment, and how that impacts the DNA. Well, it's really both. Our DNA is a set of instructions and this is providing all of the information. Set of instructions the same in every single cell. So an interesting question to think about is, how do the cells know which genes to? Well, it has to do in part of the genetics, genetic coding. So there'll be promoter regions within the DNA. They'll be meaning above and genes. And essentially what this is doing is this is creating chemical changes or tags on top of the DNA. So it's actually not changing the DNA code itself, it's providing a way that the cell can adapt to the gene, so regulate activity. And when we look at how this, a visual cue of how this is actually affecting the DNA, we can see on the left-hand side, the purple string is DNA. And what's interesting is if we were to take every single piece of DNA within our cells and we were to expand it from end to end, it would be about 2 meters long. So that's fit inside each of our cells, our nucleus, these microscopic areas within our cells. So the body has a way that it winds it up to store it. But if it's wound up, it's not accessible. So on the left-hand side, you can see there's those little chemical tags that are on the DNA itself and the proteins around the DNA, which allow the DNA to be accessible. So that means that the gene can be active. And then on the right-hand side, you can see that the information, it's not accessible. It's wound tightly. The gene itself is not, the proteins aren't able to get there. It's not able to be expressed. And so that's where we see the gene as inactive. So when we think about epigenetics and its purpose, it's a way to modify the genetic expression based on the response to the environment. So it's a way that the cell learns. It's a way that we adapt to our surroundings. And really, it's about maximizing your survival. And so the cells want to remember, essentially, the environment that it's been exposed to and how to respond to that, as well as it can be passed down through generations. So we can think about how these are kind of scars or alterations within or around the DNA. And this is typically with epigenetic changes, these are long-term changes. And so interesting, we can look at epigenetic changes in past histories where there's been famine. So women who were pregnant during the Dutch famine between 1944 and 45 actually gave birth to babies that had an increase in weight and a decrease in length. And it increased the baby's chances of having poor health about 80%. So the babies were acquiring or inheriting this metabolic skill set for starvation. And whether or not that skill set is appropriate for their current lifestyle is besides the point, because what the mother is trying to educate through the epigenetic changes is that there may be a potential for low calories at some point in the life. So epigenetics, it really helps to understand how the genetic information is being used, where it's being used in the body. So is it a nerve cell? Is it a muscle cell? What genes do we need to turn on in order to create that cell? And then when is the information being used? And we can think about the interplay between genetics and epigenetics, where genetics is really the set of instructions, our book of life, and the epigenetics is the editor. And so a lot of individuals, there's a lot of talk about, well, it doesn't matter what your genes are, it's just about your epigenetics. But we're kind of missing, we're just leaving out the whole genetic base instructions. And we find that there's a number of environmental changes that can actually affect the epigenetics within the body. So it has to do with our prenatal care. Even before pregnancy, the quality of that egg, the genetic information that's getting passed on can be affected. And so we kind of refer to this as the 1,000 days in utero, in early life, where as the baby is being developed, it can Sorry, I think there's a little bit of a mix-up there. I'll just go back.
You can just go ahead and continue, Dr. Robin. I just turned the webcam off for a moment. I think it was just freezing up on us.
Okay, sorry about that. Okay, I just wanted to make sure we're online.
Yep. Just continue.
So
we can hear you.
In these epigenetic changes, we can see that in the exposure to things like chemicals, stress, nutrient deficiencies, they can have long-lasting effects during pregnancy and those first early developmental years. We also find that there's environmental chemicals that can impact the DNA. So looking at wireless technology, which I find fascinating, especially considering the new talk about 5G wireless. It's kind of whether or not this has long-term effects, but there are studies showing that some epigenetic changes can affect neurodevelopment as well as neurobehavioral changes within children that are exposed to high levels of these wireless signals. So it has been shown to reduce memory, learning, cognitive attention, as well as behavioral problems that kind of mimic ADHD. And we're also looking at pollutants. It's kind of a toxic laden world, and we'll talk a little bit about this in the presentation later on, but we see that heavy metals, pesticides, solvents, these can have significant changes that they impart on the DNA. And even when those chemicals are removed, we can see that they can still have an effect on the DNA, how it's getting expressed. And these are through epigenetics. So we also see this with drugs. There's some fascinating research looking at how SSRIs during pregnancy can affect babies and their brain development. And then it's not all negative. So there's exercise and lifestyle we see can have really positive effects. So when we're exercising, and long-term changes can occur where there's over 500 different genes that are getting altered. So these are increasing the genes that help to prevent disease, as well as decrease genes that help to decrease the likelihood of disease. And that's really where we're interested in when we look at lifestyle genetics. When we look at doing a test like My Blueprint, we're trying to understand what are the exact changes that we can make in our lifestyle, in our diet, in our nutraceuticals, stress reduction, that can alter our DNA specifically, so our unique genetic code. Now, it may seem scary. We do know that all these effects that I was talking about can have long-term health consequences, including effects on cancer, mental health disorders, diabetes, cardiovascular disease, neurological disorders, and obesity. However, your genes are not your destiny. What we're starting to realize is there's key environmental changes that we can make that can actually alter the genetic and epigenetic modifications. So these are reversible. But it's about knowing those specific nutritional and environmental factors, as well as those gene and environmental interactions that we see. So it's exciting to have, a company like AOR, which is, bringing genetic testing to the public, and in a way that it's accessible, it's easy to understand. You don't need to be a geneticist in order to be able to understand the report or even apply the information. So when we look at a test like My Blueprint, it's really a guide. It's telling the story of how to modify gene expression specific to the individual. So we can understand where to focus our interventions. We can understand how to turn off and on certain genes, as well as alter not only short-term expression, but long-term expression. And it's identifying what we can think of as kind of those genetic weaknesses or susceptibilities. It's also interesting to look at how do we compensate for some of those susceptibilities? So how do we direct and modify the gene expression? Especially when we look at anti-aging, people are trying to slow down this process. We'll talk a little bit about how we can modify genes and their expression and the cellular environment to actually reduce the aging process. Really fascinating evidence there. And then looking at lifestyle, what we're talking about today and what lifestyle genomics is all about is what are the specific lifestyle and dietary factors that we can implement in our life in order to alter gene expression. So these are things like generally meditation, stress relief, exercise, even infrared sauna has effects on genetic expression and the health of our cells and DNA, but also we see nutrients can go in and actually act like nuclear factors to stimulate certain genes that help with repair and regeneration. Things like curcumin, green tea, even methylfolate, the active form of folic acid, and we see resveratrol as well. So there's a lot of information that we can get from a genetic test. We think about, we have 23,000 genes, and right now we know that there are very specific markers in those genes that can have very specific outcomes. So we can see how one can be affected or respond to different dietary strategies. So this is including how much fat they should eat, are they kind of primed for the keto diet? Are they sensitive to carbohydrates? And then looking at specific nutrients. So we're going to talk a little bit about this today, about phosphatidylcholine. What I love about looking at the genetics is seeing what are those nutrients that I'm actually predisposed to a deficiency? What foods do I need to eat more of? And what time in my life do I need to be aware and cognizant of some of these susceptibilities? So I can do things in my lifestyle to compensate for this. We look at mental wellness. So, are you highly sensitive to stress? What can you do about that? Food sensitivities. Physical fitness is more about repair and regeneration. So are you at risk for higher inflammation, microcircular vascular blood flow, and any injury risks as well? And then hormones, which kind of ties into how the hormones are being processed, and that's overlaps with detoxification. So detoxification, I'm going to talk a little bit more about. And the takeaway message that I want everybody to understand is that the genetic test, it's about knowing the actions to change your health. It's not about a test to tell you, what disease you're going to get or how you're going to die. This is knowledge that empowers the individual, so that whether it's the customer, the client, the patient, or just those individuals at home who are doing this test and looking for the best ways to modify their health, genetics is a way that we can help to navigate all the information that's out there. So detoxification, it's a massive topic considering how many, if we look at how many chemicals we're exposed to on a daily basis, toxic burden is a real problem. So we encounter over 80,000 different chemicals over the year. I looked this up today because I had to re-examine this number because it almost seems unattainable. But essentially what happens, 310 kilograms of toxic chemicals are produced each second. And this goes into the air, into our water, into our food. So on an annual basis, there's about 21 billion pounds of chemical pollutants that are coming from the industrial manufacturing industry. So plastics, even in farming, pesticides, solvents, heavy metals, we know these are having interactions within our body. So when we do testing, we can find at least 700 different contaminants that people generally have kind of floating around in the body. And we know that this can impact the DNA. This can actually cause DNA damage. It can alter our hormone response, our liver metabolism, and really affect a number of different systems in the body to affect disease and progression. So we can kind of think of this, because it affects so many different areas, they're generalized symptoms that we see. It could be something like fatigue and headache and weakness. Just, you go to a doctor and you see these things, there's hundreds of different diagnoses that it could be if you try and break it down and find the underlying cause. We know it has an effect on the immune system. So increased susceptibilities to colds, kind of generalized inflammation that creates a lot of joint and muscle pain, and then food or environmental and chemical sensitivities. And what you'll typically find is it'll be acquired. So as you continue on in life and you may not have had allergies as a child, but you notice as you get into the latter decades of your life, more and more sensitivities kind of pop up and they're unexplained. And overall, this is creating inflammation and oxidative stress, and that's what's damaging the DNA. And when we damage DNA, it's putting more demand on the cell in order to try and repair that DNA. And this is kind of the inner working mechanisms that lead to aging and what can contribute to more of that rapid aging for individuals. So the important piece is, rather than kind of wait for all these symptoms to come up, you really want to find out who is susceptible to toxic burden before the symptoms arise. And this is where we can look at the genetics and see some of those pathways that are responsible for breaking down those chemicals. So this is actually from my report. And on the left-hand side, you can see there's a little take action. And that take action, it's telling us that there's a change in the genetic code that is altering how I process certain chemicals. And so it's giving me an indication that I have an increase in sensitivity to what's called polyaromatic hydrocarbons. So these are found in well-done meats, in smoked meats, cigarette smoke, pollution, and I'm actually processing them where it increases the risk of oxidative stress. And so this is where you want to be particularly mindful in where you're getting these chemicals, as well as how you're preventing the damage that can ensue with these chemicals and their exposure, and how can I recover and kind of protect the DNA. And this is where you can get into supplementation. So there's things like milk thistle, helps with protecting the liver from any sort of damage with chemical exposure. And then sulforaphane actually will increase the phase 1 detoxification pathways. Resveratrol and N-acetylcysteine, good antioxidants, has been shown to reverse oxidative damage to DNA and help compensate for some of these issues. So then when we look at how this affects kind of aging overall, I think this is where you can get into some really cool kind of novel ways and ingredients that we can start kind of manipulating the genes directly. So a product that I'm really fascinated about, it's called Benagene. And Benagene has been shown to stimulate over 350 anti-aging genes. And it's really about increasing our mitochondria and increasing our glucose metabolism within the cell to increase ATP, or energy production. And this has been shown to help increase the health as well as the lifespan in animals. And it regulates blood sugar levels and mimics caloric restriction. So caloric restriction, when you look at studies with humans, it can delay the disease process related to things like kidney disease, autoimmune disease, diabetes. It reduces neuron loss in neurodegeneration like Parkinson's and Alzheimer's disease. So it has a quite interesting, effect on the body overall. And so if we can mimic that, that's pretty exciting. It's a powerful antioxidant, as I mentioned, can support the anti-aging. So Benagene, it's oxalo acid, but it contains something called 3-carboxy-3-oxo-propanoic acid. A little bit of a mouthful there, but we just call it Benagene for obvious reasons. And so what's interesting is it is a natural compound. It is thermally stable within the AOR product. And what it's doing is kind of inserting itself within the energy production. So glycolysis as well as mitochondrial respiration, and is showed to increase energy production and alter that redox balance. And this is just a diagram to show how the calorie restriction, when they look at certain studies where they did calorie restriction, what genes were being expressed, and then the effects that the oxaloacetate had, there's actually an overlap there where you see over 350 genes that are in common with these effects. So you can see it increased lifespan. This was in worms, and it's activating certain genetic pathways that is altering the genes in order for cellular benefit. So we can think of mitochondrial function, mitochondrial support. It's important for brain. So ALS, as well as brain cancer, some clinicians have shown benefit using this. And then when we look at kind of animal studies, we see that the actual mitochondrial biogenesis, so ATP production, energy production, it affects signaling with the insulin pathway, neuroinflammation, and can really help to maintain neurogenesis and prevent from that neurodegeneration. So pretty exciting. And just to kind of summarize too, so weight loss, when you look at energy metabolism, it actually prevents the body from storing excess fat. And then it can actually prevent and repair DNA damage, which is thought to be part of this aging process. So it is a very strong antioxidant. And this is what we want to be mindful of when we're thinking of finding who is susceptible to DNA damage. What are some of the ways that we can help compensate for that and protect the DNA overall? So another part of gene expression has to do with how the genes, how changes in our genes are affecting our risk for nutrient deficiency. And one vitamin that I want to highlight is choline, because not many of us really think about choline. Even in my practice, it's not something that comes to the forefront when I'm looking at patient health. But what we can do in a genetic test is we can look at a whole slew of vitamins that we may or may not have a good lab test to really reflect tissue levels or whether or not there's a deficiency. So with choline deficiency within itself, damage. And what's interesting, it's men and postmenopausal women that are at increased risk for choline deficiency. And the reason for this is that estrogen is responsible for increasing the expression of this gene. So there's less risk in premenopausal women when there is higher estrogen levels. And then when they transition into postmenopausal or perimenopausal, that's when they're at risk for liver damage, and it ends up increasing fatty liver deposits. So non-alcoholic fatty liver disease is related to choline deficiency. We can also see an increase in muscle damage as well as risk for cardiovascular disease. So I'll go through some of the mechanisms of this, but with cardiovascular disease, choline is one of the main pools that we use our methylation donors. And so if there is poor what's called endogenous production of choline, then it's going to be using up a lot of those methyl donors and creating a tendency for high homocysteine, which can affect cardiovascular health. And then when we look at fetal development, choline is particularly important for brain and neurological development within the baby. And you think, well, choline, it's found in the diet, it's in eggs, it's in liver, but that doesn't mean that everybody's getting sufficient amounts, even from a generalized standpoint. So there's studies looking at women in California that they, over 25% of these women were, their intake of choline was under the amount that they should to prevent birth defects in their babies. So it's important to identify these women, identify who is at risk, and kind of understand some of these pathways of how we can alter them, what timing of someone's life that we need to kind of intervene and prevent. So this is just kind of a quick diagram for those visual learners. At the top there, we can see our internal processes of how we get choline in the body is through the conversion of phosphatidylethanolamine. This is getting converted into phosphatidylcholine, and this is done by an enzyme called PEMT, P-E-M-T. So there's a genetic alteration that we can test for that will show us how well this enzyme is likely to function. So some individuals, they have a decrease in that conversion of the PE to the PC. And if the body is low in phosphatidylcholine, it can affect bile. So this is affecting our fat absorption. It's affecting our excretion of solvents from the liver, through the bile, and can contribute to liver disease. It also affects bile motility because bile is important for stool, for bowel motility. So it can affect constipation. We also look that it's an important molecule for signaling within the membrane. So insulin resistance, obesity, liver disease, as well, it's an integral part of our cell membrane. So it's allowing the cell membrane to be nice and fluid, to allow the receptors to be available for hormones and other transcription factors to bind or proteins to bind. And without this healthy kind of fluid membrane, that's where we can see a lot of diseases start to ensue. And then at the top there, you can see its relationship to homocysteine. So as the PE is being converted into phosphatidylcholine, it's using up one of our methyl donors to add to the homocysteine pool. So high levels of homocysteine will cause cardiovascular disease. So by identifying just this one gene, this is just one example, we can see how it affects so many different areas within our health. So there is a product called citicoline, which is CDP choline, so cytidine 5-diphosphate choline. This is interesting product because there's a lot of clinical research looking at CDP choline in particular and its effect on neuralgia. So nerve pain, nerve inflammation. We see that CDP choline helps with the body's own production of phosphatidylcholine. So it's adding to that pool of available phosphatidylcholine where it can get directed and converted into all those areas that I showed you. So it can help with neurotransmitter production. That's one area in that diagram I would have added, is that phosphatidylcholine helps with acetylcholine, which is for memory, for nerve function. It's improving cellular membrane functions. So as I mentioned, supports the nerve, the brain, and the eye. And also our own production of phospholipids within the cellular membrane. So this is important because we don't want to disrupt the balance, the natural balance that the body needs. And we don't know, if you're supplementing with a high amount of, say, phosphatidylserine, we really don't know how that's affecting the overall balance within the cellular membrane. And we want to make sure that we're not potentially depleting any other phospholipids. And as I mentioned, when we increase the body's own production of choline, it helps to reduce homocysteine. So overall, when we're looking at genetic testing, how this is being utilized, we can see that it can help to predict who is susceptible to environmental toxins. So we're looking at prevention. We want to make sure that we're identifying who's susceptible, removing those chemicals, and overall, this can help with a number of different diseases. But what's interesting, we can look at it kind of in the anti-aging realm as well. And when we look at kind of identifying where do we start when we're coming up with a protocol to help individuals, or you have symptoms, they're kind of generalized, where do you start? You want to make sure that you're including kind of the right product for you. And you don't do that unless you have some objective data that is helping you to understand, where these products are working and what's going to be most effective. So really looking at kind of the right individual product for the right individual. We're really avoiding kind of a trial and error approach, especially when it comes to weight loss. I alluded to the fact that genetic testing can impact or reveal some of the areas, whether we're sensitive to fats or sensitive to carbohydrates, how to kind of modify our macronutrient ratios, and this can affect our weight loss efficacy. Rather than trying a ketogenic diet and seeing how it works for a couple months, why not get some of that objective data to understand how you're responding specifically and then modify the diet accordingly. And we also see this in effects for losing or decreasing cholesterol, triglycerides, exercise effect on diabetes. All of this can be predicted from a genetic test. So it's really valuable information. When we look at chronic symptoms, say you've been trying everything, going to a number of different doctors, doing kind of a lot of blood tests, nothing is coming back with any sort of answers, and you're not getting improvements, there's something that may be missing there. So it's about kind of identifying this next step, this next layer of information, which can help to understand some of the disease processes and how to address it. And so choline, in addition to vitamin A, essential fatty acids, B12, iron, all of these come highly from animal sources. And so if there's a vegan or vegetarian out there, it's especially important to identify whether there's a risk factor for these deficiencies, because what we see is these deficiencies won't necessarily be a problem until the diet is devoid or low in these areas. So you're finding out who is susceptible and then the appropriate compensation. So there's supplementation, there may be ways or alternatives to find other ways to get these vitamins as well. And I talked a little bit about pregnancy, just those 1,000 days preconception, pregnancy, and then early development is so crucial when we look at DNA development. So overall neurodevelopment, neurobehavioral development, and the effects on the genes and epigenetics during that time, which can really affect someone's, life, whole and entire life thereafter. So I think it's important to know as a mother, prior to conception, are there ways that you can maximize certain nutrient stores, prevent certain susceptibilities, and just be really mindful about maybe some of those risks to toxin accumulation, which we know can have a big effect on baby's health. And there are some family diseases that we look at. So although this isn't a disease-based genetic test, it's not genetic-based diseases that we're looking at, there are a few that are lifestyle genetically based. So diabetes, how you're responding to carbohydrates, same with metabolic syndrome. Hemochromatosis is a genetic disease, but this has to do with iron overload, so it's very modifiable of how this gene and this disease presents. These would be the individuals you want to avoid iron supplementation. So something as, harmless as iron can be actually extremely damaging for those who are carriers of this or present with this. And they could be a carrier, meaning that they can pass it on to their kids. So we also look at heart disease and then celiac disease too. So what's interesting is these are all modifiable by the environment. So again, it's that knowledge that we're accumulating from this test that we're able to then make very specific recommendations. And this is all part of what we call precision medicine, so where we're looking at a predictive value of this test and then determining the precise interventions that are going to help to compensate for these genetic alterations as well as prevent disease. So in summary, we talked a lot about, the epigenetic and genetic modifications. How are we turning genes on and off? And it's done through the environment, looking at, the in utero and early stages of development, how toxins play a role. Certain drugs can have a big effect on our genetics. And what we want to tap into is that lifestyle and dietary strategies, the nutraceuticals. Because these epigenetic changes can have long-term health consequences, but it's not our destiny per se. These epigenetic changes can be reversible. There are ways that we can compensate for changes within our genes, within our lifestyle, as we talked about with detoxification, removing some of those chemicals, or even adding in certain products that help with energy metabolism, help to repair the DNA and reverse some of that DNA damage. And then this is helping, as we talked about, dietary factors that we can implement as well. So looking at who is at risk for different nutrient deficiencies, how do these nutrient deficiencies affect our symptomology, our disease progression that we may not have thought about, or we're not able to test. It's hard to test for something like choline deficiency. So this is really starting to bring kind of these topics to the forefront of the mind to say, when am I going to be susceptible to risk of deficiencies? So is it during pregnancy? Is it, postmenopausal for women related to choline? And when am I going to have to kind of be more cognizant of where am I getting this from my diet, or perhaps supplementation as well. And so we talked about a little bit who benefits. So it's prevention, it's anti-aging. We talked about pregnancy, pre-pregnancy, vegetarians, vegans. I would add in, family history of disease. I think there's something for everybody. And overall, I really think that this is going to be something in the next 5 years that it's going to continue to develop, continue to kind of explode into the public domain as people and healthcare practitioners become more and more adept to looking at the genes, how to affect it, and kind of integrate that into a full protocol. So that's all I have for today, guys. Sorry for little disruptions there, but thank you so much for joining me today and listening. if there's any questions, I think now is the best time to write them in the sidebar there. Thanks.
Thank you so much, Dr. Robin. This was a really, really informative presentation. We do have a couple questions, so I've answered a couple in the question bar, but if anybody wants to submit, I'll be taking a look at them. But I thought there was one question that was asked that I think that would be great to get your take on. The question was, does my blueprint tell you if a particular gene is expressed or active or not?
Yeah, that's a great question. I get that all the time. And we're really looking at the code. So we're looking at what is the actual instructions, what are the actual letters that are written out within the DNA. It's telling us what the potential is for how that gene is getting expressed, but it's not telling us how it's getting expressed. So it's not about the expression, it's more about what is the potential. What does that say? You know, we can think about it's like a library. So the books are all there, all the information is there. The next step is to understand how are these genes getting expressed, you know, what books are we taking out, and how that's affecting health overall. So it's a very astute question, and that's where we look at some of these symptoms. And what you'll find in my blueprint is it'll say, do you have symptoms of iron deficiency? Follow up with your healthcare practitioner, you know, if you do, and look at iron status. And so this is where we combine the genetic testing with other kind of objective markers to make sure that we understand how this is affecting health overall.
Perfect, thank you. Is there any sort of testing that is now being done to look at that kind of protein side expression of things? Can you comment a little bit on that?
Yeah, this is exciting. It's proteomics, metabolomics. There's epigenetic tests out there. About 6 years ago, I went to a conference and they were talking about kind of this massive AI machine that was in development. And what it would do is kind of test 5,000 different data points. So including the genetics and all of these protein expressions. You can look at mRNA, you can look at protein levels, you can look at enzyme function. There's a whole slew of information that is out there. However, that accessibility, the cost effectiveness, we still have some time, I think, in development. There are epigenetic companies as well. Again, it's about 1,000 to 1,500 pounds that it costs to look at this, and they would expect you to do that kind of biannually or annually. So I think the price point will come down eventually, but for now, I think it's very useful to kind of look at the genetics and then work with a healthcare practitioner and just see, you know, how that's getting expressed.
Perfect, that makes sense. A follow-up question on the first one that I asked is, you know, you talked about if genes are becoming active and are expressed. Is it possible to reverse this once the gene's kind of been turned on? I think you kind of talked about this, but if you want to say a couple more things about it being turned on and off, and if we can actually manipulate that.
Mm-hmm. Well, there's a few different kind of concepts here that we're talking about. So genetic expression, separate from epigenetics, we can think that we understand and we know that our genes are getting turned on and off, sometimes thousands a second, because the body is very smart as far as it doesn't want to waste any energy. So it's not going to increase the expression of a gene. Let's use an example of vitamin A. So your body isn't going to use the energy to make the vitamin A enzyme.
We have a little bit of those tech issues. I'm going to go ahead and turn off.
That, unless your body is requiring that in the
Dr. Robin, you there still? Okay, I'll answer a couple questions while we wait.
We'll find that those genes get turned on, the necessary cells, and then utilize it. In epigenetics, so this is where I was talking about those chemical tags that can occur.
Okay.
And those chemical tags, they can be long-term, and this is where it can affect generations. Some of these chemical tags we're starting to understand can be reversible, and that's where we're starting to look at some of these epigenetic environment gene interactions. But the answer is yes and no. So they are reversible to some extent, while others we know are getting turned on because of the environment. So exposures to chemicals, certain nutrients, disease processes, and that's how we kind of start to break this whole picture down and know where to look when we go through some of these genetic tests and panels.
Okay, thank you. A question about supplementation and the way it manipulates your genome and the expression. Will it act as kind of a band-aid, or is this something that it can actually take off some of those chemical tags you were talking about?
Well, that's why I use Benagene as one of the interesting examples, because we were actually able to see that it can repair DNA. It acts like an antioxidant, as well as stimulating some of those antioxidant pathways and genes related to aging or anti-aging. So because of that metabolic effect that it's having, it's turning on those genes. So it is compensating for certain individuals who have an increased risk for DNA damage. That would be a perfect supplement that I would include for people who have poor glutathione pathways, poor detoxification, their mitochondrial function may be decreased, maybe their methylation is altered. You're starting to see multiple pathways that are responsible for protecting our DNA and alleviating some of the toxic burden on the body to help kind of compensate for this. And then we look at, there's this whole other topic of nutrigenomics, which is talking about the products like curcumin and green tea that actually go and they will signal DNA transcription factors. So they'll promote the expression of certain genes. So say, for example, curcumin, it increases our glutathione enzymes and can kind of decrease our cytochrome phase 1 enzymes. So it's an interesting effect that it's having, where it's helping to kind of slow down phase 1, but speed up phase 2. And that's where we start to see some of the I wouldn't say band-aid effects, but it's really altering how the genes are getting expressed to kind of compensate for these inborn tendencies that we have genetically, and how that can help mitigate some of the downstream effects that we see in the health overall.
Perfect. I think that gives us a lot more clarity in that. And those are very commonly asked questions. I know you lecture about this all the time, and I'm often there with you. Those are things that people are asking. So those are excellent questions. We did have one question about, does genetic testing tell us about chromosomal defects? Not this type of genetic testing. That's a different type. That my blueprint will not cover that. So there's no pathological association with any of the genes that we're looking. That's why we're focused on making it a lifestyle and nutrigenomic testing. And then finally, there was one question about acetylcholine. How much time does it take to have a beneficial effect to address disease? And I can answer this one. There's research on acetylcholine using it usually a little bit more longer term. So you're looking at more of a 3-month window. That's often how it was used in a lot of the studies. Much of the research on acetylcholine comes from Europe, Eastern Europe, and also Scandinavia and Spain. And it does take time for the fatty acids to be incorporated into the cellular membrane, to have its effects on blood flow, to have its effects on nerve function. So we have to give it some time for it to work. Thank you so much, Dr. Robin. I think that's all the questions that we had. Those are excellent questions, and you presented excellent information. If anybody has any other additional questions, you can email them to marketing@aor.ca. Again, that's marketing@aor.ca. Our next webinar is coming up on February 11th at 12 p.m. Eastern. Dr. Aaron Zedek, a naturopathic doctor, with a focus in sports nutrition, we'll be looking at that topic, optimizing athletic performance. If you're not registered yet, you can sign up at AOR/B2B webinars /. Thank you so much for taking the time to join us today, and have a wonderful afternoon.